The filtration membrane. Glomerular filtration is a process in which water and some solutes in the blood plasma pass from capillaries of the glomerulus into the capsular space of the nephron. To do so, fluid passes through three barriers that constitute a filtration membrane: the fenestrated endothelium of the capillary, the basement membrane, and filtration slits. The endothelial cells of the glomerular capillaries are honeycombed with large filtration pores. These are highly permeable, but small enough to exclude blood cells from the filtrate. The basement membrane consists of a proteoglycan gel. The basement membrane excludes molecules larger than 8 nanometers. Even some smaller molecules, however, are held back by the negative charge on the proteoglycans. A podocyte of the glomerular capsule is shaped somewhat like an octopus, with a bulbous cell body and several thick arms. Each arm has numerous extensions called pedicels that wrap around the capillaries and interdigitate with one another. The pedicels have negatively charged filtration slits that are about 30 nanometers wide, creating an additional obstacle to large anions. Almost any molecule smaller than 3 nanometers can pass freely through the filtration membrane into the capsular space. This includes water, electrolytes, glucose, fatty acids, amino acids, nitrogenous wastes, and vitamins. Such solutes have about the same concentration in the glomerular filtrate as they do in the blood plasma. Some solutes of low molecular weight are retained in the bloodstream. However, because they are bound to plasma proteins, they cannot get through the membrane. For example, most calcium, iron, and thyroid hormones in the blood are bound to proteins that retard their filtration by the kidneys. The small fraction that is unbound, however, passes freely through the membrane and appears in urine.